So Monday, Plymouth Bluff, I'll remind y'all of that on Monday morning. We'll do our plant collection. So far, everything is set where we're going to do that. And I'll have lab set up so y'all can get in and practice for the midterm. Okay. So I believe that we finished up. Oops, gotta start the share before I do that. We finished up all the bryophytes. Do y'all have any questions? I'll take that as a no. How are y'all doing with the material so far? I do want to hear from y'all, okay? Because when we ask those questions, if we get no answer, we assume y'all were just fine. And okay, just want to make sure. Okay, good so far for Kyra. Okay. I suggest making a table. Just like we did with the algae, you want to start, you want to start making a table for the different bryophytes. So compare the bryophytes. Can anybody tell me a difference between the liverworts and the mosses? Think of anything. Okay. Brandy, so leafy structures. Can you be more specific? Who has leafy structures and who doesn't?
Okay, if I remember right, it's the liverworts have leafy structures, and then only some mosses do, I think.
It's the opposite.
Okay.
So think about the phalloid liverworts, the ones that are like onto the ground and they kind of dichotomously split. But there are some leafy liverworts. And then all of the mosses have phyllidia, the leaf-like structures. So good thoughts, just we got those backwards, and that's an easy fix. Okay, so you want to think about that. Think about something very specific to the hornworts. If you ever hear anything about having a basal meristem, basically, if you're in the bryophytes for sure, you're talking about hornworts because they have that continuously growing meristem at the base of the sporophyte. Okay, so this is the purpose of the tables, is to try to identify those features that are going to help you tell one from another. And interestingly, if you may not realize this, but if you were to go further into like systematics in biology, which is a study of, well, it's kind of the study of relationships based on characteristics, DNA or morphological. I'm basically setting y'all up with morphological characteristic comparisons and kind of getting you thinking about like, so what is the feature I need to know to separate this from that? What feature is it that I need to know to separate this other thing from this other thing? What features are common between these two species or two groups? Okay. So that's the purpose of those tables, and to try to get y'all thinking about relationship. Okay, plant of the day is actually 3 plants. Poison oak, poison ivy, and poison sumac. Most people's favorite plants, right? Some of y'all may not be allergic. That's okay. I'm glad for you. I think I'm kind of just a little bit allergic. I don't have big bad rashes. There are some people that it seems like they can just hear the name poison ivy and they break out. I do think there are some people that are so allergic that if you're near the plant, some of the oils are a little bit aerosolized and can affect some of those people. You may have heard the term poison oak. I grew up hearing the term poison oak, and it was not referring to this plant. It was actually referring to a Virginia creeper, which it's a misnomer. It's the wrong name for the wrong plant. Poison oak will have 3 leaflets. Virginia creeper has 5, and I was always told that poison oak has 5, poison oak ivy has 3. That is not true. I do think we've got poison oak in Mississippi. I think I have seen it. Poison ivy, on the other hand, we have it everywhere. So it's probably the most common of the ones that you see. I will try to point out some of this to you when we go out to Plymouth Bluff to help you avoid touching it. So it is an interesting plant. You can see some real features here. The bottom right is poison ivy. The one on the left is poison oak. And the one up here is poison sumac. I have not seen poison sumac in Mississippi. That's not to say it's not here. It's just that I haven't seen it. It's not common for me. And I can show you other sumac, but sumac is not the same thing as poison sumac. They're in different genera. So if someone says sumac, don't go, oh no, be careful. No, that's fine. Actually, there's some sumac that are quite edible. It's the poison version. You have poison sumac at your house, Elena? Are you sure it's poison sumac? 100%? Can you get pictures? I'd love to see it. Yeah, if you can get some pictures, I'd love to see it. Because a lot of people do get sumac and poison sumac mixed up. And if I'm not wrong, poison sumac is viney, whereas other sumacs are like little trees, kind of bush-like structures, and that's maybe one way to tell them apart. So if you do see them right now, the other sumac has these big groups of little fruits that kind of dangle sometimes if they're older. You'll see them kind of pointing up. So many people get those confused. But I don't know if that's a cool thing or not to say that you've got that at your house, Elena, because that's not fun for your mom. Okay. One way to see poison ivy, at least in the winter, when you don't have the leaflets to look at, are these kind of hairy looking vines. It's not hair, these are adventitious roots that allow them to attach to trees and other structures. Here is the dermatitis that can result. I've never had anything this bad that I can remember, maybe as a kid. Some people will get it and cover their body, and it's not good. It's itchy as I'll get out. There are other vines out there that you might see besides poison ivy, and you'll notice that, like here on the left, you can see the vine portion, not the tree that it's wrapping around, not having those hairy kind of adventitious roots. This one wraps around, and notice how it can actually, affect the growth. It can actually kind of almost girdle the growth of this these small saplings. And cool enough, you can actually get something that you can make these really cool walking sticks from. So, but this one's Lonicera japonica. Can anybody tell me what the common name of this one is? Anybody recognize it? I'd be surprised if no one recognized it. Flowers taste good. You don't eat the flower though, you eat the nectar.
Honeysuckle?
Yeah, it's Japanese honeysuckle specifically. Yeah, this one. The vine version is the Japanese honeysuckle. There are some bush honeysuckles like Amur honeysuckle. But yeah, it's honeysuckle. I like that one. It is an invasive and quite annoying, just so you know. But here's some interesting uh, walking sticks. Some of those obviously are not affected by vines, but I just threw it in there. Okay, so let's describe the next group that we're going to get into. In fact, this group is every plant from here on out. Okay, the tracheophytes are going to include the next three major groups of plants. So we've talked about the bryophytes. We have three more major groups. We have basically the ferns and fern allies. We have gymnosperms, the pine trees, things like that, and then the angiosperms, the flowering plants. So we are about to get into all three of those, and that whole group has the name tracheophytes, also called the vascular plants. And from within the vascular plants, we think that all of them probably developed from some common ancestor that was probably a moss. And the development of true vascular tissues are what separates this group from the mosses. We now have xylem and phloem, hence the reason for why they're called tracheophytes. So because of the true vascular tissue, we have the development of roots, stems, and leaves. There are two groups. If you want to, you can also like divide it into two groups, even though I just said there were three here that we're going to talk about, but you can divide it into two more groups. There are the cryptogams. They are the groups that are the ferns and fern allies that do not produce seeds. And then the other two groups that we talked about, gymnosperms and angiosperms, fall into what's called the phanerogams. And the phanerogams are those that have seeds from sexual reproduction. So the distinction between these two things are no seeds and seeds. Everybody good with that? Okay. So some of the problems that we see solutions to are that, again, we're still not in water, but we're going to move further away from having to live right up against the water like we see in the bryophytes. Remember, the bryophytes are living maybe on a rock in a creek, in a wet cave, on the side of a building that has constant water or very, very frequent water. And that helps to get the sperm to the egg, for example, for replication, for reproduction. Now here we see a desiccation resistance in the spores. The sporopollenin that I mentioned comes up again and again. We see it in the spores. We'll also see it in the pollen later on after we get to flowers. Because we want the spores that are produced, especially when we're talking about the ferns, to not dry out as it moves from one location to another. Here is one of the major differences. Now I'm emphasizing this, one of the major differences between the bryophytes and the rest of the plants. Here we have a large sporophyte generation. It's now the dominant version, whereas in the bryophytes, it was the gametophyte that was the larger. Of course, we expect to see an epidermis and cuticle, no different than bryophytes. Here we've got very well-developed guard cells and stomata, so we've left behind air pores back in the bryophytes. And they have an upright habit where they've developed a system, a shoot system that has stems and leaves. So what I mean by upright is that they're not laying on the ground like this anymore, absorbing light. They're doing this. What do you think that three-dimensional move, that shift, has done for the plant? What's the benefit? Catch more sunlight. Um, exactly. Think about the coleus that we saw the other day in lab, and we noticed that the leaves were oriented kind of opposite each other, and then it would shift when it went to the next set, and then shift back and forth going up the stem. The whole purpose was to orient the leaves so that it maximized the amount of sunlight. Being three-dimensional moves you upwards. You now can produce leaves outwards and get lots more light, as opposed to just being that on that two-dimensional surface. You're now a three-dimensional organism. Exactly. Now, ultimately, we're going to see the development of a root system. Roots, not necessarily the first thing that you get when you're looking at tracheophytes, and we're going to see something that doesn't have roots or leaves, just a stem in the ferns and fernalii group. And I have a few in the greenhouse, hopefully still alive. I'm having some problems with the watering system. We see xylem and phloem, not surprising here because this group is the tracheophytes, the vascular tissue, vascular plant group. So obviously they've got to have vascular tissue. And of course, this is one of the reasons why they're able to be three-dimensional and stand up. They now have that mechanical support that you see with xylem and phloem. Also, that allows for more nutrients like water and mineral nutrients to move upwards and things to move downwards when you're in the phloem. We see more protection of the gamete, and we see more protection of the spore-producing structures, which is starting to mean that we're getting internalization of some of the sexual reproduction processes. So they're being protected inside the parent organism. This means that you now don't have the need for water as a reproduction medium. You don't have to have water to get your sperm to the egg. Okay, that's not necessary anymore. The bryophytes needed that. It's not necessarily the case after this. You also see the development of heterospory. Does anybody know what hetero means? What does the prefix hetero mean? Any ideas? I know y'all know this, somebody. It means different. Yep, exactly. So what do you think heterospory means? Different spores. Okay. So what we're going to see is the development of some plants that will go from just producing a general spore to producing a you can maybe think of it as a male spore and a female spore. Okay, we're going to call it micro and mega spore later. So this is what leads us to being able to produce seeds eventually. By the way, the plant that only has stems and no leaves or roots is the one pictured on the right. Okay, now remember tissue systems. We've got the dermal, the ground, and vascular tissue systems. Keep these things in mind because they're going to come into play as we move on from here. The dermal is the outer covering, so it's what makes your epidermis or your periderm. You'll also see the endodermis in the root. Epidermis is primary in origin. Cork, which is produced in the outer part of the trunk for trees, starting to help with making bark, is secondary in origin. The ground tissue are all tissues that are not vascular or dermal. They surround your vascular tissue, and these are things like your cortex and your pith. And then, of course, vascular tissue is what makes up your xylem and phloem, and that's a complex tissue because there's more than just tracheids and xylem vessel elements, sieve tube elements. There's more than just those. There's also some parenchyma and other things in there. Okay. So we've got to focus on this vascular tissue because we're in the vascular plants now. The word stele, S-T-E-L-E, is the vascular cylinder that we see moving up the plant, or cylinders. And there is a pattern of these in the primary vascular tissue that you'll see in stems and roots. These patterns are important to know. The way that you see xylem, phloem, and some pith involved are going to give certain looks to them. So the first one, protostele. You know, proto means first. It's the most ancestral. There is no pith, and it has a leaf trace, but no leaf gap. And you may go, well, what does that mean? A leaf trace is when leaves are formed off the side of see if I can draw, because I'm not sure if I can describe this. So here's your stem. And of course, eventually we'll get this where we'll have some leaves that come off the stem. See if I can change the color. I'm gonna do yellow. So we've got vascular tissue running upwards. When it branches over into the leaf, maybe down the midrib, this is a leaf trace. So that vascular tissue is kind of like this. It's connected. There's no gap in this formation of the vascular tissue. Okay, so protostele looks like, I'm going to draw at the top kind of a very simple one. So here is the very center, and I'm going to just kind of color it all in. I can't do a very good job at all of this is vascular tissue. Okay, there is no pith in the middle. It's just vascular tissue. Y'all good with that? Just somebody shake a head if you're okay with it, hopefully. Okay. Siphonostele is maybe the next one that would have developed. It's a little bit more complicated. Siphonostele has a pith, and it's one big singular vascular cylinder, but it has a leaf trace and a leaf gap. So if you were to draw this from a cross-section perspective, it'll look like this. So here's the outer part of your stem. It makes an O shape. And I, for the life of me, can't draw with a mouse. Okay, so this is vascular tissue. This is pith. Okay, and y'all can see that here in this left picture. What's the difference between the other one and this one? There's a pith here in the middle. The other one had no pith. It's just vascular tissue. So leaf trace and leaf gap. You can see the leaf trace here in this picture. Leaf trace moves upwards, but there is a gap in the next part of your vascular tissue. Okay, and you can see how there's a, and here's another picture off to the right. So remember, we're talking about a cylinder, right? You'll see what I'm talking about? See my hands? Here's a cylinder. It's like this. There are places where it goes up, and of course, the cylinder separates. Okay, and you have a gap. That's what we've got here. Why do we have a gap? Because the vascular tissue that would have gone straight up, continuing the stele, ends up coming outwards into the leaf. Everybody good with that? But that doesn't mean that it breaks completely, because remember, it goes all the way around. So part of that cylinder continues off to the sides. So we've got a leaf trace coming out into your leaf. The gap is right above it. It will reform eventually, especially, by the way, think about this. It's solid cylinder during the node section of the stem. It's at the inner node that you get the leaf trace, leaf gap picture. That makes sense for everybody? Okay. Down here in the bottom right, you can actually see how you've got vascular tissue and then pith in the middle. So that's an example of what it would look like. Okay, thank you all. Okay, now we've got one more. We've got the eustele. And when you see the term EU, it means true. Okay, like eubacteria are the true bacteria. Eustele, this is the true stele. This is the one that y'all have already seen. Have y'all noticed that you've got a cross section and then you've got vascular tissue like this? Does this look familiar? You've got this concentric ring of vascular tissue and you have pith rays that separate those, don't you? Okay, so the first one to develop is your prostele, and then you still get that solid cylinder of steles and the siphonostele. And by the way, think of siphonostele, siphon, siphonostele, siphon. I think of it as a straw, and a straw is a cylinder. But then over here in the eustele, it's a bunch of separate, distinct bundles of vascular tissue. So you've got all kinds of leaf traces and leaf gaps as each one of those. Kind of like you may have one going up together and the other one veers off onto the leaf. Okay. Before I get to the root, do y'all have any questions about stele types? It's going to matter as we get into the different kinds of plants, because as you develop some of the early types of plants, there are, okay, some of the early types of plants don't have this kind of stele. We see either a protostele or a siphonostele. So it's a good characteristic to know, oh, okay, that's an early plant. Okay. Now, this also affects what you see in the root. So right now, what I've described is what happens in the stem. Okay, these are stele types in the stem. Then we're going to get to the root. So the origin of the root is that we don't see embryonic formation of that place where the root starts, the root axis, in some ancestral plants. Basically, roots are developed from a type of stem. Eventually, we started to see stems that we would call rhizomes. They start to get vascular tissue. They start to develop underground, and they start to be horizontal. So we see that in some of the early plants with early types of roots. Notice that in those early roots that are horizontal, we see protostele like anatomy. Okay, so protostelic anatomy. So what is protostele? Tell me this, just those of you that can see me, or if you can type really quick, or I should say those I can see of you. Does protostele have a pith? No, it does not. Okay, so we're going to see a slightly different look to the vascular tissue in the root than we will in the stem. So what you might notice is that, well, if protostele developed first, and if it was during the time when most plants had protostele that roots developed, and roots developed from stems, what we're seeing is that roots developed from stems that were protostele, and they've kept that feature into the future, at least in some cases. Okay, even if they're above ground parts, the stem, are not protostele. Okay. We see leaves developing eventually off these stems, little protuberances that develop on the shoot, and we see these are part of the shoot apical meristem. Leaves themselves don't have meristems. I don't know how many times as a kid I might try to put a leaf in a thing of water and try to get it to grow roots, and it usually didn't work. And a part of that is because the meristems are not something that are there. There are two kinds of leaf anatomy. There is the microphyll, which is ancestral, and the macrophyll, which is the derived feature, the newer feature. And microphyll and macrophyll um, are slightly confusing terms because we are going to see those terms used two times, and they're going to mean a little bit different things depending on the species what we're looking at. Okay, so here we're talking micro and macro, meaning micro small, macro big. Okay, later we'll see the term micro and macro, meaning male and female. Okay, or at least in general what that means. So, but here it's small and large. Microphylls are very small usually, and they have a single strand of vascular tissue running through the middle. You can see that here. So basically, when you look at this vascular tissue, it's protostele. Okay, no pith. We see this in the group of called the division in Lycophyta, which we'll go into more detail later. They're the only group of tracheophytes. I shouldn't say only. For the purposes of this class, it's basically the only group of tracheophytes with a macrophyll, maybe one other, okay, that we may mention. So I'll try to bring that up when we come into it, the exception to this kind of rule. But the reason I point this out is that division Lycophyta is the one I want you to think of. When you think of microphyll, think Lycophyta, okay? Now, the origin of leaves, there is one theory that it's called the innation theory. And innations are little nubs on the outside of a stem that are really nothing. Exactly, they're just, I don't even know what you call it. They're just little protuberances that kind of developed on the outside of the stem. What we'll see is something like this. I'm going to try to draw this progression down here. So an innation might look like this. No idea how that happened. See if I can erase it, because it's not. Okay, so that's an innation. And here's your vascular tissue. Okay, so early on, we have some plants that don't have leaves. They just have these little bumps. Later, we'll see the development of stems that have this happen. With some of this vascular tissue kind of starting to work its way into these little protuberances. Here we don't have anything going in it, but here it's kind of trying. Over here, it'll eventually put vascular tissue fully into something that we would call a leaf. Do y'all see what's going on there? This is called vascularization of lateral appendages and what will basically become leaves. Does that work for y'all? Okay. Macrophylls, as opposed to microphylls, are usually large. They're complex and they have a lot of branching. You can actually see the branching of the vascular tissue in this leaf up here with lots of vascular tissue. And usually you'll see siphonostele or eustele in the macrophylls, as opposed to the protostele in the microphyll. All tracheophytes, except Lycophyta and Psilophyta, and maybe one other group, have macrophylls. So let me ask you this. Is microphyll versus macrophyll a useful feature if you're talking about a difference between Lycophyta and any other plant? Absolutely, okay, because it's the only one that has that feature. Xylophyta, on the other hand, doesn't have anything. It just has enations. It doesn't have leaves of any sort. Now, the telome theory of origin, as opposed to the enation theory that I just showed you, is more of this idea. So it's a little more written out. We get a branching system like we see here in this maple leaf through a system called overtopping, planation, and webbing. Overtopping, where you see some telomes leave the stem behind. So telomes in this case are going to be some of the vascular tissue that will kind of branch out from the stem. Kind of like what you saw with the enations. We started to see a little bit of protuberance in the vascularization. But you don't just get one straight line of vascular tissue. You start to get branching, and that's what's happening with the overtopping. Eventually, what you see is a flattening out into a certain plane of that those telomes, and then in between, you get the development of parenchyma tissue that we see that makes up the blade of the leaf. So those are two origin theories that people have tried to figure out, like how leaves developed. And it's possible that both of these are true. The enation theory is probably what developed a microphyll, and then maybe this is what developed a macrophyll. And it looks like macrophyll probably was the better bet for most species. So, okay. I won't go into much detail asking you about those, enation theory and telome theory. What I will want you to understand is that enations are little bumps that are not vascularized. And later we think that maybe enations started to develop vascularization and maybe later some branching. So, but the idea is that enations came first and then leaves, as with whichever theory the case may be, we got leaves developing from vascularization of some sort of little protuberances that push their way out of the stem. Okay. I know this is not the most exciting portion of botany. I know that. Okay, now something important, not necessarily fun to talk about, but it's extremely important. You want to make sure you get this. Ding, ding, ding. I don't know how to emphasize this anymore. Okay. If you want to star this, highlight this, write this as a note on your forehead, whatever you need to do, we've got to make sure you get homospory and heterospory and understand how it works. Okay, homospory. Homo means one type, okay? Same. What we see in the terms of making spores through meiosis, remember, sporophytes are 2N. We get a 1N spore. You have to go through meiosis to do that. In making a single type of spore, a homospore, okay, this is called homospory. We see this in ancestral tracheophytes. They make one type of spore, and the gametophyte that develops from that spore is bisexual. Okay, it's able to produce gametangia that make either male or female gametes. Homospory is found in all members of Xylophyta, which we're going to see, Sphenophyta, some Lycophyta, and some Pterophyta. Why are these important? Because these are the four major groups that we're going to talk about in the ferns and fern allies group. Okay, so all of Xylophyta is homosporous, all of Sphenophyta is homosporous, some of Lycophyta is homosporous, and some of Pterophyta is homosporous. One type of spore. Heterospory develops later. This is the, remember, precursor to making seeds. Later, we get development of vascular plants that make two types of spores. We've got the microspore that'll be produced in microsporangia on the sporophyte, and they will make development of microspores that develop into microgametophytes. So basically a single type of a gametophyte that only produces sperm. And then you've got the megaspore, the female, which produces is produced within megasporangia that make megagametophytes. So these will be your gametophytes that develop from spores, and these gametophytes will only produce eggs. Okay, so you've actually got two different life cycles, okay, two different portions of the life cycle that are not on the same plant, one producing male, one producing female parts. We see heterospory, so heterospory is usually how you do say that. So heterospory in Lycophyta. Now, not all, some of them are homosporous. There are some in the Pterophyta. It's the minority of the group, but there are some. And then every seed plant is heterosporous. Okay, all seed plants, all gymnosperms, all angiosperms are heterosporous. In the production of microspores in the microspore mother cells or microsporocytes, we produce lots and lots and lots of microspores. Okay, we'll see that in large amounts. However, when we're talking about megaspores, one megaspore mother cell will produce 4 megaspores. One microspore mother cell will produce lots and lots and lots and lots of microspores. But one megaspore mother cell makes 4 megaspores. So kind of keep that in mind. Hold on. I want to see where we're at. We can discard that. I want to see how far we are. Okay. I think I can get these 3 slides in the last 4 minutes. Okay, homosporous plants, those that make one type of spore, the gametophytes are large, okay? Even though they're not the dominant of the life cycle, the gametophyte will still be large, and it will be independent of the parental sporophyte. So remember back in the mosses where you've got your gametophyte, the green leafy part, and then you've got that capsule looking thing that sporophyte produced off of it? They're attached. Here, they're going to be separate from one another. They're going to live completely separately as separate plants. The gametophytes might even be photosynthetic because they have to live separately. They've got to be able to produce their own food. Some of them even connect to endomycorrhizal fungi. So a way for them to live is to create a symbiosis with a fungus that helps to provide some food and water. This is relatively short-lived, but it happens. The embryo sporophyte that is produced, remember, those gametophytes end up a lot of times having the spore, develop inside it and then kind of grow out of it. A lot of times what happens when these are separate is that the embryo sporophyte that's produced, because it's not being taken care of much from the parental gametophyte, is that it dies a lot. Okay, there's a high mortality rate for the poor little sporophyte embryos, and it's because you've got exosporic development. The gametophyte is independent of the parent, so because they're relatively separate from one another, they don't get a lot of protection. Gametophyte to sporophyte, sporophyte to gametophyte. Heterosporous plants, on the other hand, are gametophyte dependent, especially nutritionally, on the parental sporophyte. So you see what's called endosporic development. It's not free living. Over time, we see a continual progression of change in size of the gametophyte. It gets smaller and smaller and smaller while it's being protected inside It will eventually get to the point where the gametophyte will be 3 cells. That's it, 3 cells if you're a microgametophyte in the flowering plants, and 7 cells with 8 nuclei in the megagametophytes. And we're going to describe that in lecture 2. We'll see some in lab. Not today, but I mean, not obviously not today, but not soon, I should say. But we will see this. So what will go from being a visual plant that you could see, multicellular, it will go down to very, very, very small amounts of cells. And some other things that we'll see is that the archegonia in all cryptogams and phanerig, we see archegonia, which produce eggs, and all the cryptogams, which are the ferns and fern allies, and all of the phanerogams except for the flowering plants. So when we get to actual flowers, we won't see an archegonial structure. Antheridia is in all cryptogams, and then we don't see it again after that. All the seed plants don't have an antheridium. And we're going to see flagellated sperm still in some groups. We'll see it in all of the cryptogams, even if it's not necessary. And we'll see it even in some higher plants, the cycads and the ginkgos, even though it's not necessary. So it's just an ancestral trait that's been stuck around. You might even want to call it vestigial if you want. So what we see is this progression towards the form of living where you use seeds to produce your offspring and their way of moving around. And it's the ultimate adaptation of life on land for plants, at least at this point. Okay. Now, if you're like, I don't really get all this, maybe take some time and kind of ruminate it on over the weekend, and we'll talk about it more on Monday. This is a general outline of kind of the basic ideas of what's happening in the next 3 groups of plants. Okay. So if you're like, I didn't really follow that, we're going to take it and go in depth and try to illustrate every one of these points with examples from here on out. Okay. And that's all I've got for today. Y'all have a good weekend. If you've got questions, let me know. I'm going to go start setting up a practice exam. I'll send you an email when it's open and free, and you can come in this afternoon, maybe even later this morning if I'm able to get it out as soon as possible. Okay. And that's all I got.